Thank you so much for joining us today for this Lena Biosciences webinar. My name is Jenna Gallegos. I'm from Samba Scientific, and I'll be the moderator today. In this webinar, Dr. James Shoemaker from Lena Biosciences is going to present data on novel assays and services for studying drug-induced liver injury.
So, good afternoon. I'm Dr. James Shoemaker, and I'm the Chief Science Officer at Lena Biosciences. Lena Biosciences provides drug discovery platforms with human-relevant data for the development of safe and effective medicines. In this webinar, I will introduce our proprietary assay services, which capture an early toxicity signal and then enable a systematic study of that signal to understand the origin of toxicity and its relevance for drug-induced liver injury. Before I present the outline, I would like to take a moment and highlight the general principle of Lena Biosciences' approach to the DILI screen. We use a battery of assays to probe the cell to find out what triggers the toxicity. These assays measure different cell functions. The function that gives the lowest IC50 is the toxicity trigger, because that function is lost first, and the cell cannot compensate for it. The assays then track which other functions fail as the dose increases. For a given drug, the trigger mechanism is the same for all patients because the same function fails. However, the final impact for the individual patient may be different based on the cascade of events following the trigger and the ability to adapt. The reason why we know that the same trigger function fails is because that function fails even in donors with no known genetic risk factors for that function. To achieve this, we developed fit-for-purpose assays which detect the loss of function at a very low dose, so that the patients with risk factors can be identified early. So what is the practical impact? Our approach provides a better understanding of the relevance of the liver signal. It provides the origin of toxicity in the liver, highlights how the toxicity progresses, where it finally leads, and what the consequences for the patient are. Our approach may be able to identify early the patients who are at a particular risk, thus reducing the risk of late-stage drug failure. Our approach may contribute to better decision-making, especially in early drug development, before significant investments are at risk. Here's the outline of the webinar. I will briefly highlight the DILI problem, explain our approach to identify what triggers DILI, show how our primary screen works, show how our secondary screen works, explain the trigger mechanism for example drugs, and show that while the trigger is the same for a given drug, the outcomes can be different for individual patients. So what is drug-induced liver injury? Drug-induced liver injury, or DILI, is a condition where the liver sustains damage as a result of exposure to medications with sometimes severe consequences, which can be fatal. DILI is a major problem for patient safety and a grand challenge for the pharmaceutical industry and regulatory agencies. Why is DILI such a big problem? Because it is detected late. Analyzing the endpoints does not solve the DILI problem. It helps with the detection and diagnosis, but none of this sheds much light on where and how the problem started. For this reason, drug-induced liver injury is difficult to predict. Intrinsic liver injury happens to everyone when the drug dose is too high. Idiosyncratic injury is patient-specific. It is usually detected in post-marketing after the drug is approved and used in a wider population. Clinicians classify DILI as intrinsic or dose-dependent and idiosyncratic or dose-independent. We believe that everything is dose-dependent the first time the injury occurs, but thresholds can be lower with re-challenge. Why? Here's an example. For many drugs, phase 2 transferases are successful in detoxifying the parent when the dose is low. When the dose is high, they become saturated and a higher portion of the drug undergoes phase 1 metabolism. Many phase 1 reactions are oxidative, which may contribute to the formation of reactive metabolites and free radical intermediates. Reactive metabolites can form covalent bonds with proteins, lipids, and nucleic acids. These adducts are sometimes recognized as antigens which can activate the immune system. Depending on how the activation proceeds, some reactions can be immediate allergic reactions, while some can take weeks or longer if T cells are activated. Thus, as far as the trigger is concerned, everything is dose-dependent the first time. Upon re-challenge, even a very low dose can cause havoc, because adaptive immunity has memory. In sum, the immune DILI trigger is the same for everyone, but patients can develop idiosyncratic or mixed DILI, but most do not develop DILI at all. Once DILI is triggered, however, how the toxicity evolves and where it leads can vary. The same drug can cause different kinds of liver injury in patients. Thus, the best way to approach the DILI problem is to look for the trigger to be able to prevent it. What can we conclude? We can conclude that the offending drug caused the injury, but the outcomes were different in patients. To solve the DILI problem going forward, we need to find and understand the origin of the first DILI signal to be able to recognize it in the future. What is our approach? Our approach is to find the trigger. The trigger tells us where and how DILI started. What is the trigger? The trigger is the first cell function that fails. Now, this is very important. Once the first function is lost, we may be at a point of no return if the cell cannot compensate for that function. In other words, the drug has liver liabilities for all patients. Because the trigger is the first function that fails, we need a good model and very sensitive assays to detect that. And this is what we do. The purpose of our primary screen is to find out where the trigger is located in the cell. To find the location of the trigger, we use a battery of assays that sweep through different cellular compartments. The assay that gives the lowest IC50 tells us in which cellular compartment the trigger is located. Here are a few examples with triglydzone, diclofenac, and isoniazid. The arrow indicates the assay which tells us in which cellular compartment the trigger is located, because that assay gives us the lowest IC50. For many toxic drugs and drugs with box warnings, the trigger can only be found in the first dose. In other words, it takes one dose to lose a critical cell function. Therefore, to find the trigger compartment, we dose once and read at 24 hours. So what is the goal of our primary screen? To find where in the cell the trigger is. Now let me take a moment to explain these graphs a little bit better. Each line on each of these graphs measures some cell function. This is called a dose response because we are monitoring how a cell function behaves with an increasing drug dose. The vertical axis is a measure of a cell function normalized to vehicle or control without the drug. For example, a value of 0.4 indicates that 40% of this function is maintained by the cells, but 60% of the function has been lost. IC50 is the drug concentration in vitro for which cells experience a 50% loss of function. We are looking for the function that gives us the lowest IC50. We refer to that function as the trigger. Cmax is the maximum drug concentration in human plasma, both bound and unbound. When we divide the IC50 in vitro with Cmax in vivo, we are attempting to estimate at which multiple of the human dose we see the problem in vitro, or in our trigger case, the first loss of function. If the ratio of IC50 to Cmax is equal to 1, that means that our model is very good because we can predict the toxicity in vitro at a dose that is given to humans. As you can see, for triglydzone, the ratio of IC50 to Cmax is below 1. This indicates that the first loss of function happens at a fraction of the therapeutic dose. Indeed, triglydzone was withdrawn from the market. If the ratio of IC50 to Cmax is high, for example 20, but we know that a drug is toxic to a small group of patients at Cmax, we ask ourselves, what is the relevance of the signal we see in vitro? In other words, is our model good enough to predict the toxicity observed in humans at that dose? Because we were able to predict the toxicity of withdrawn drugs close to Cmax and those which cause idiosyncratic DILI in a small group of patients at an IC50 over Cmax less than 25, we believe that our model and our assays are relevant for the drugs we tested because we can fast forward to the actual patient outcomes in just one day. So once again, what is the goal of our primary screen? To tell us where in the cell the trigger is. But is there something else that the primary screen tells us? There is. The primary screen also tells us how the toxicity evolves and which new mechanisms of toxicity become activated as the dose increases. What else do we notice? The likelihood of tolerance or adaptation to the drug insult is drug type dependent. For isoniazid, some functions may be able to tolerate or adapt to the drug even when the dose is high. When we see a situation like this, where some functions are maintained and the majority have failed, we suspect that the drug will cause delayed toxicity, at least in some patients. Indeed, isoniazid causes primarily idiosyncratic DILI. Once we know where the trigger is located in the cell, we run a secondary screen in that cellular compartment. The purpose of the secondary screen is to find out what the trigger is. In other words, which function fails first in that compartment. This is an example with protein kinase inhibitors. We are showing two assays in the most likely trigger compartment. The potential trigger for ceritinib, crizotinib, and ruxolitinib is mitochondrial complex 1 inhibition. The first three drugs, sorafenib, ceritinib, and crizotinib, have warnings and precautions for DILI. Their IC50 to Cmax ratio is less than 10. Ruxolitinib does not, and we can see this from our data because the IC50 to Cmax is 19. Ruxolitinib is also less likely to inhibit transporters compared to the first three drugs. Our mitochondrial toxicity assays are holistic. They are sensitive to everything that happens inside the cells to the extent that it affects mitochondria, which includes drug metabolism and the inhibition of active transporters. Sorafenib and crizotinib have reports of fatal DILI. Ceritinib does not, but ALT was up in 28% of 925 patients, according to the package insert. For patients taking ceritinib, once-a-month monitoring of ALT, AST, and total bilirubin is required. Ruxolitinib does not have warnings and precautions for DILI, but the clinical trial listed in the package insert lists elevated ALT in 25% of patients and elevated AST in 23% of patients. Our IC50 to Cmax for ruxolitinib's mitochondrial toxicity is 19.3. Currently, our internal empirical IC50 to Cmax cutoff is 25. Thus, we would classify this drug as DILI positive. Many protein kinase inhibitors are toxic and used for severe and progressive diseases such as cancer when the benefits outweigh the risks. Our secondary mitochondrial toxicity screen is sensitive enough to detect sorafenib's toxicity at a fraction of the dose given to patients and to correctly differentiate between the more toxic sorafenib and the less toxic ruxolitinib. How much can be saved if toxic drugs fail early and fail cheap? A lot. 23% of drug withdrawals were due to hepatotoxicity. Can we help to predict high-risk drugs and raise the patient safety standards? The FDA analyzed 1,279 drugs. 60% of the drugs were found to have a DILI risk. When DILI is detected in post-marketing, the FDA puts a box warning. Can we predict which patient groups are at risk before the drug is approved? Can we predict which patient groups are at risk before clinical trials? Can we stop toxic drugs from progressing to first-in-human trials? We are answering that question in a dish every day. And here is how. If we want to assess vulnerabilities for a given dose for the first-in-human trial, we need to pick a dose and go bottom up to see which cellular functions are at risk of failing. Now, let's run triglydizone's hypothetical first-in-human trial using its therapeutic Cmax as a dose. What do we see? We see that two cell compartments are struggling. We have a complete loss of function for the light green dose response. This is the mitochondrial compartment. And we have a 40% loss of function in the dark green dose response, which is a separate compartment. So what does this tell us? This tells us that the dose is high. We have at least two active mechanisms of toxicity, one with 100% and one with 50% loss of function. The dose should be much lower and/or a different formulation considered. Based on this hypothetical first-in-human trial in a dish, triglydizones is a no-go. However, triglydizones was approved. 66 patients died. Can we predict who was at risk? The patients who were at risk were those who were struggling to maintain the highlighted functions prior to the drug challenge. Who else was at risk? Patients taking another medication for the same indication, or medications which had mitochondrial and transporter liabilities. Patients with cholestasis were at high risk because the triglydizones metabolites were eliminated via the bile. Patients taking other medications which were eliminated via bile were therefore also at risk. Who else was at risk? The groups which were traditionally at risk. How do we know this? We are detecting the toxicity at Cmax using the cells from seemingly more resistant individuals. So we just revealed that the major mechanism of triglydizones toxicity at Cmax is mitochondrial toxicity. BISAP inhibition is close, but secondary to mitochondrial toxicity. This tells us that all ATP-dependent transporters are also at risk because they need mitochondrial ATP to pump metabolites, bile acids, and conjugated bilirubin outside the cell. The majority of triglydizones metabolites are eliminated via the bile, and the bile salt export pump, or BISAP, depends on mitochondrial ATP. This means that the metabolite elimination is likely impaired due to mitochondrial toxicity. In other words, this toxic drug inhibits its own elimination. We tested the activity of BISAP, MRP2, MRP3, and MRP4. Triglydizones and/or its metabolites inhibit the transporters at a slightly higher dose than the IC50 for mitochondrial toxicity. Our transporter assays are also holistic. They account for drug metabolism, reactive metabolites, free radicals, mitochondrial toxicity, and everything else that a cell is dealing with when we measure the transporter activity. Because reactive metabolites can bind to and inactivate functional proteins like BISAP and enzymes like transferases and mitochondrial proteins, it is difficult to tease out what happens first, but we believe that we have found the right order. Now, let me reiterate that our hypothesis is that triglydizones inhibits its own elimination. If that is true, then the end result is that metabolites could be accumulating in the liver and that triglydizones concentration could be rising in the blood. Triglydizones is lipophilic. Lipophilic drugs can passively diffuse into cells and cross tissue barriers. We could have had mitochondrial liabilities elsewhere. To check this, we tested triglydizones in our in vitro brain model. The model does not incorporate a blood-brain barrier, but we know that triglydizones can cross it. Triglydizones caused mitochondrial toxicity at its therapeutic Cmax. The IC50 was only 3 times higher than in the liver model, but there were differences. The major mechanism of mitochondrial toxicity was not the same. So what does this tell us? Again, the dose was high. Who was at high risk? Patients with vulnerable mitochondria and those who had cholestasis and did not know it because 97% of metabolites were eliminated via the bile. Who else? Patients who took medications eliminated via the bile. So where does this lead? A widespread cellular energy crash. Why? If tissues which rely on oxidative phosphorylation, such as the brain, have mitochondrial toxicity, they will demand more glucose from the liver, and the liver cannot meet this need. It is busy dealing with all the problems I outlined earlier. Plus, triglyceride inhibits hepatic gluconeogenesis. The end effect is probably a cellular energy crash for most patients. Is there a hypothetical pattern? We have not yet done enough studies to know this for sure, but based on 4 drugs tested, we know that triglyceride caused mitochondrial toxicity in the brain and the liver. Pioglitazone did not have mitochondrial toxicity in our liver model, but we have not yet checked the brain. Fenformin had mitochondrial liabilities more prominent for the brain. Metformin had mitochondrial liabilities for the brain, but not for the liver. So what does this tell us? Tissues which depend on mitochondria for energy production may be at risk for certain types of drugs, but the final outcome for these types of drugs likely depends on the liver. What I have just outlined is merely a hypothetical exercise that we can use to develop a decision-making tree. How good the tree is depends, of course, on the quality of our data. Triglyceride was the first in-class glitazone to treat type 2 diabetes. It was withdrawn from the market after 3 years and 66 reported deaths, but it was not indicated as initial therapy in patients with type 2 diabetes and had a warning for hepatotoxicity. The warning stated that transaminase should be checked at least monthly during the first year and at least quarterly thereafter. Idiosyncratic reactions were also listed. Triglyceride's box insert stated that the primary route of excretion was in feces. What does this tell us? Triglyceride was primarily eliminated via the bile. Today, triglyceride would most likely be caught in the in vitro drug-drug interaction studies. Many of our colleagues in the in vitro space have also shown that it is toxic. So what can we learn from triglyceride? Which drugs could have similar risks? Those with significant elimination in the bile on top of mitochondrial toxicity caused by both the lipophilic parent and the reactive metabolite. In terms of the parent, reactive metabolite, and BISAP inhibition, tolcapone fits this profile, but has lower fecal excretion. Entacapone does not have a reactive metabolite based on the literature, but its excretion is 90% fecal. Both drugs were tested using a single assay from the primary screen, the one which identified the trigger for triglyceride. Both drugs came back positive for the liver and brain liabilities. For both drugs, the IC50 for mitochondrial toxicity was below that reported for BISAP inhibition, suggesting that ATP reduction is the cause and BISAP inhibition the effect. Because we don't look at mitochondria in isolation, anything that happens intracellularly to the extent that it affects mitochondria is reported by one of the probes in our primary screen. However, we assayed only one function and thus cannot say what the trigger is. Tolcapone was withdrawn and later introduced with careful monitoring of liver enzymes. It has a boxed warning. Entacapone has a precaution for patients with hepatic impairment and post-marketing reports of adverse events characterized as hepatitis with mainly cholestatic features. With only one assay used, our IC50 to Cmax predicts that tolcapone is toxic at a fraction of a dose given to patients. Entacapone is less toxic, but still DILI positive based on our empirical IC50 to Cmax less than 25 criterion. So, once again, what is our DILI approach? Find the trigger. Fast forward to patient outcomes and learn with evidence. So how did we learn? When we started, we knew what our platform excels at, and we developed and validated the first assays. One of the first drugs that we tested were fenformin and metformin. We were not impressed, but we learned. First, we learned that pH can have significant effects. Second, we learned that we needed to better we needed better assays to catch toxic drugs that do not have significant P450 metabolism. Third, we learned that there may be a pattern to the data, which is the most important point of this slide. Diclofenac and isoniazid were also in the first batch of drugs which we tested the assays in both the liver and brain model. And we did that for a good reason. Liver is the major site of drug metabolism where we could have bioactivation. Brain is the site of higher oxidative metabolism and oxidative phosphorylation, but less extensive drug metabolism. What did we learn? First, how the toxicity evolves with increasing dose can be very different for the liver and the brain. Second, if the liver fails to detoxify the parent, the brain is at risk because these drugs can cross the blood-brain barrier. Third, the likelihood of tolerance and adaptation is organ and drug dependent. So what is the purpose of this slide? The patterns are different with different classes of drugs, but relatively similar for drugs within the same class. Now let's see what the primary screen told us about isoniazid, the most commonly used medication for tuberculosis, which has a boxed warning for severe and sometimes fatal liver injury. The screen isolated an unusual faulty function as the most likely toxicity trigger. I say unusual because this function is not on the list of the usual DILI suspects, but we always thought that it should be. The signal in the liver came from both the cytosol and the mitochondria. The signal in the brain came just from the cytosol. Brain mitochondria were less affected than the liver mitochondria. Were we confident that the trigger was in the cytosol of the liver cells? No, we needed to test earlier. What was interesting is that the liver model still had a chance to recover, while the brain model did not. Do we think that we can catch all toxic drugs with one dose and a 24-hour readout? No. This is why we define the domain of validity. We do not expect to detect the toxicity of drugs which take time to reach steady state, change protein expression over time, affect nuclear or mitochondrial DNA replication, etc. These compounds require longer time testing, but we do know that we are most likely dealing with one of these compounds when we compare a 48-hour readout with a 24-hour readout. In these instances, we need to dose at least twice. If the trigger is in steady state, we will catch it. If it is not, it becomes much more expensive and more difficult to find it because we need to run the assays at different time points to identify the loss of function. Now let's talk a little bit about our assays and what makes them different. On the left is the Seahorse XF assay for mitochondrial toxicity using triglycerin as a model drug. On the right is the triglycerin-pioglitazone mitochondrial toxicity assay by Lena Biosciences. What makes our assay different? First, our assay does not measure cellular oxygen consumption. Second, our measurements are not based on the serial injections of an ATP synthase inhibitor, an uncoupler, and the inhibitors of the mitochondrial electron transport chain, together with the drug, to isolate the mechanism. We do not use any uncouplers, inhibitors, or any other drugs to isolate any mechanism of toxicity, be it mitochondrial toxicity or something else. While it is tempting to do that, we found early on that it was very difficult to draw conclusions due to unpredictable drug-drug interactions, and to be even more specific, metabolite-metabolite interactions. Our assays are clean. Our probes and reporters are clean. We assay after the drug has completed its action. We are not using the glucose-galactose assay, but we do appreciate the simplicity and the low cost of the galactose assay to isolate mitochondrial toxicants. Our assays are whole cell-based assays. They are run in 3D cell cultures grown in our platforms. We are not using substrates used with isolated mitochondria. We are assaying the activity in whole cells, which can make reactive metabolites. Thus, all of our assays measure indirectly the effect of reactive metabolites to the extent that these reactive metabolites affect the functions in the cellular compartment in which we are assaying. Mitochondria have many vulnerabilities. Indeed, a compound can interfere with the electron transport chain, the citric acid cycle, mitochondrial DNA replication. It can uncouple oxidative phosphorylation from the proton gradient, induce the mitochondrial permeability transition pore opening. Our secondary mitochondrial screen focuses on specific functions. We have 5 validated assays, but I'll be just showing 2. Here's an example with nifazodone, trazodone, and buspirone. What do we see? Nifazodone in red is more toxic than trazodone, which is in blue. Trazodone in blue is more toxic than buspirone, which is in green. The domain of validity for this statement is based solely on the potency in the mitochondrial compartment. Our keystone complex 1 assay is more or less targeted to mitochondrial complex 1. It captures mitochondrial complex 1 liabilities both directly and indirectly. Directly, when complex 1 is a sole target, and indirectly, when complex 1 is inhibited most often as a result of another downstream complex inhibition or problems with the citric acid cycle. However, keystone complex 1 will also report the effect of transporter inhibition, or anything that happens in the cytosol, to the extent that it affects the mitochondrial complex 1. Our keystone complex 1 to 5 assay captures liabilities in complexes 1 through 5. It measures both direct effects and also indirect effects induced, for example, by mitochondrial transition pore opening, or anything that happens in organelles, the cytosol, or the plasma membrane to the extent that it affects complexes 1 through 5. Keystone complex 1 to 5 is a broader assay than keystone complex 1. If the only liability in the electron transport chain is complex 1, we would expect that complex 1 dose response would be more sensitive and give us a lower IC50 than does the complex 1 to 5 dose response. We can see this for 2 of the drugs, trazodone in blue and buspirone in green. As you can see, the solid lines for complex 1 function are to the left of the dashed lines for complexes 1 to 5 function. For nifazodone, complex 1 to 5, the dashed line, gives lower IC50 than complex 1, the solid line. Thus, for nifazodone, there appears to be at least one more liability in the electron transport chain, or with ATP synthase, in addition to complex 1 liability. Indeed, a target validation study in isolated bovine heart mitochondria found that nifazodone inhibits complex 1 and complex 4. This is what we see in our data. Complex 1 to 5 function, the dashed line, is to the left of complex 1, the solid line, because it captures the liabilities in all complexes, not just complex 1. We then looked closer. Our IC50 for complex 1 was higher, 18.8 micromolar, than that reported by the target validation in the bovine heart mitochondria, which was 14 micromolar. We deployed our second probe for complex 1, also known as keystone complex 1b assay. That assay gave an IC50 of 11.3 micromolar. However, the liability in all complexes was overall 7.9 micromolar. Why? Are our assays wrong? No, they are not. Our studies are done using whole human liver cells, which contain mitochondria. The target validation was done in isolated bovine heart mitochondria. So why are our values lower? The answer is reactive metabolites. All of our assays indirectly capture the effect of reactive metabolites. One of those metabolites appears to be a strong inhibitor of either complex 2, 3, 4, or 5, but not complex 1 directly. Does nifazodone have reactive metabolites? Yes, it does. Nifazodone has numerous reactive metabolites. Still, do you think that bioactivation always implies that the reactive metabolite will cause toxicity? The answer is no. The potassium cyanide and GSH trapping assays are performed in NADPH supplemented microsomal incubations to capture reactive metabolites. They can tell us if reactive metabolites can form, but not if they can cause harm. First, a cell has to make enough reductants, such as NADPH or NADH, to make these metabolites. Second, assuming that it does, only a whole cell assay can tell us if these metabolites can cause harm. Many drugs have structural alerts and reactive metabolites, but they do not cause any problems. Why? Most often because of the dose and pharmacokinetics or formulation. A low dose can, but not always, reduce the risk of bioactivation. In sum, bioactivation and the formation of reactive metabolites contributes to nofazodone's mitochondrial toxicity. The trigger is the loss of function of at least one of the complexes 2 through 5. So nofazodone is a no-go. In the US, nofazodone was withdrawn in 2004. It was later reintroduced because it can be effective in patients with resistant forms of severe depression. It has a boxed warning for hepatotoxicity and can cause serious liver damage and death. Trazodone could be an uncoupler, but that is not all. It also most likely inhibits ATP synthase directly. However, target identification in bovine heart mitochondria does not show this. Why? You are guessing correctly. Trazodone has metabolites which can be problematic. Trazodone is less toxic than nofazodone based on our assays. Indeed, it has reports of hepatobiliary disorders in post-marketing, but not a boxed warning. Lastly, our IC50 for buspirone was over 5 times higher than that reported by target validation in bovine heart mitochondria. Can you guess why? Buspirone's metabolites are less toxic than buspirone itself. Most metabolites are inactive, except for one, which has a potency that is a fraction of that of buspirone's. The metabolism of buspirone may actually serve as the aha moment for drug safety associated with reactive metabolites. Why? Because buspirone's major metabolite in humans is impervious to further CYP3A4 oxidation to the reactive quinone-imine metabolite. The absence of the quinone-imine reactive metabolite explains why buspirone lacks idiosyncratic toxicity, despite decades of clinical use compared to its structurally similar nofazodone. Because buspirone's metabolites are less toxic than buspirone, our IC50 is lower than that reported by target validation in bovine heart mitochondria. In other words, we are capturing the effect of the parent and the metabolites, not just the parent. Buspirone is not a concern based on our assays. It is used as a control, non-cytotoxic agent in the assessment of other psychotropic drugs. Antidepressants are hotspots for DILI risks. According to an FDA study, 86% were DILI positive. So what can we conclude? With one dose and in one day, we were able to fast forward to patient outcomes with high sensitivity and excellent dynamic range when differentiating toxic and non-toxic structurally similar drugs in this class. Our IC50 to Cmax range is 1.8 for the toxic and over 26,000 for the non-toxic drug. I presented the secondary screen using mitochondrial toxicity example for a good reason. The analysis of the FDA's adverse event reporting system for DILI found that mitochondrial toxicity is 1.43 times more likely to cause DILI than other mechanisms of toxicity. There was a higher mean patient age among the reports, suggesting that age may play a role in susceptibility to idiosyncratic DILI via mitochondrial mechanisms of toxicity. I just mentioned that older patients may be at a higher risk for mitochondrial toxicity. These patients also typically take more than one drug. Collectively, this puts them at a higher risk for drug-drug interactions and DILI. For most of our studies, we use either a 10-donor pool of primary human hepatocytes or commercial cell lines. The 10-donor pool models the response of an average person. If a function fails in an average person, we know that it will most likely fail in a susceptible patient. We never had to use a single donor except one time, when we suspected low P450 activity in the 10-donor pool. When developing new assays and troubleshooting, we use cell lines to cut costs, but we qualified them and we know what they can and cannot do. In 2020, the IQ-MPS affiliate to the IQ Consortium published a paper with a DILI training set for microphysiological system providers like us. We tested the majority of the drugs on that list, except for pemeline, which requires a license, mipomyrcin, which was cost prohibitive to test, and phyluridine, which mechanism is outside of our domain of validity for one dose. Many of these drugs were tested with just one assay, and we still detected the toxicity of toxic drugs at a low dose and correctly differentiated those which were safe. Our internal empirical DILI cutoff is an IC50 to Cmax ratio of less than 25. We use that cutoff to classify DILI-positive drugs. One drug, levofloxacin, has post-marketing reports of adverse DILI events due to hypersensitivity, but came back clean in clinical trials with over 7,000 patients. We tested it for only one mechanism, trovafloxacin's trigger, and it came back negative. Cross-reactivity of the hepatic injury between different fluoroquinolones has been shown in a small number of cases. For example, a patient who had a reaction to ciprofloxacin can develop a reaction to levofloxacin. We thought that we would not be able to detect that in a random 10-donor pool, but we predicted that levofloxacin was less toxic than trovafloxacin. And that was true until yesterday. We tested a second assay, and levofloxacin came back positive. The IC50 to Cmax is less than 10, and probably actually around 2, but I've not had a chance to update the table yet. So thank you so much for your attention, and I'm ready to answer any questions that you may have.
So we have a couple of questions about the length of the study.
Okay.
So have you ever done longer studies? What's the longest study you've performed? And then conversely, have you tried time points at shorter than 24 hours to assess the kinetics of your assay signal detection?
Sure, absolutely. Good question. So in terms of the length of the studies for these drug toxicity studies, where we're trying to identify these vulnerable compartments and what the cause of the DILI is, we found that it's actually most effective at these shorter time points. However, other studies that we've run, just looking at tissue health as we evaluated our perfusion system as we were developing it, we've grown cultures in our system for a month or more. So that's the longest study that we've run. Now, we did run into cases where a drug that we tested was so toxic at 24 hours that we really couldn't generate a good dose response curve. So in that case, that's when we would go to earlier time points. So we'd go to 12 hours, 8 hours, 4 hours, things like that, to really get a look at how that toxicity changed over time.
Got it. Thank you. And does your system support immune-driven toxicity detection? So
in the way that we run our assays currently, it does not. We do not incorporate Kupffer cells into our model. However, that can be done. And so if that's of interest to anybody, we'd be happy to work to develop that model in our perfusion system. Absolutely.
So a few more questions about the models. We discussed liver and brain. Do you have any other organ models that you can use?
Yes. So again, similar to what I mentioned before, for most of our drug testing, we focus on liver because of the site. It's a site of drug metabolism. Brain because of its high oxidative phosphorylation and dependence on mitochondria. So they're quite vulnerable. However, for other purposes, we have previously developed models with lung tissue. We've done primary cancer tumor tissue. And really our system is amenable to any type of cell type that you want to grow. So the neuronal model that we use for toxicity is fairly simplified, but we have previously generated more complex models that comprised neurons, astrocytes, and microglia, so we could have a more reactive tissue.
And is the system amenable to testing biologics as well as small molecule drugs?
Yes, absolutely. So again, we've focused on small molecules because of that published list of drugs that we wanted to use to validate our system. But absolutely, you could test biologics in the system, but those experiments would inherently be longer because of the longer dwell time that they have in the body.
So we've talked a lot about safety. Do you also offer services for testing drug efficacy?
Yes, we don't routinely do that, just in our own internal studies, but we have had customers who are interested in looking at the efficacy of a drug. And so, as I mentioned, let's say something for lung cancer or something like that. We have our lung cancer model, and we can test for on-target efficacy. And then if you have a healthy lung culture as well, we can make sure that it's not going to have off-target effects as well. So yes, absolutely.
And then in terms of both safety and efficacy, what's the turnaround time for your testing services?
So typically, as I said, it's the assays that we run are fairly short because we can really capture that toxicity early. But for a particular contract, depending upon how many drugs and how many replicates and different what kind of donors that they want to be used, it's typically 2 weeks or less.
So this next question has a couple layers. So you mentioned that the cells in your system are cultured in 3D. Is the sensitivity that you're able to achieve dependent on the fact that they're cultured in 3D? How would that change if there was 2D culture? Mm-hmm. And then how does your system compare with other commercially available microfluidic models that are considered semi-3D?
Sure. So yes, we have tested in 2D. We have found that the way that we culture our cells in our 3D system is significantly more sensitive. If you're just looking at IC50s, we get lower IC50s using our system. So I did not have time in this webinar to actually go into how our model works, but we incorporate a secret sauce that we refer to as our synthetic hemoglobin, which allows for increased oxygen availability to the cells in the culture. And so we culture our cells in 3D in scaffolds that we commercially offer, and they are in an insert plate that has the synthetic hemoglobin underneath the cell culture medium. It's not mixing with the medium and it's not touching the cells. It's just acting as a reservoir of oxygen, similar to how hemoglobin functions in the body. And what that does is it allows for on-demand oxygen supply for the cells. So rather than them becoming glycolytic, as they typically do in cell culture, they can use their mitochondria more effectively. And so when you're looking for mitochondrial liabilities, if the cells have the alternative or they're completely dependent upon glycolysis instead of the mitochondria, then you may miss those kinds of things. So our system is not a microfluidic system. It is, we have a tube that pumps this liquid in and out of this tray. I'd be happy to see Dan asked this question. We could speak offline and I could go into more detail as to how this works. It was just a little bit beyond the scope of this webinar.
Awesome. Thanks, James. So regulatory agencies are certainly pushing for less animal testing. Can you talk about how your services and platform are contributing to those efforts, and maybe whether or not you're speaking with regulators, and also how it compares to other in vitro options?
Yep. So absolutely, that's becoming a big push now that the requirement for animal testing has officially been lifted. It's not required for every single drug to do that. This is a huge opportunity for microphysiological system providers such as ourselves to show how effective these tools can be at de-risking drugs. Because the issue with animal testing in general is that an animal is not a human. And as drugs become more human specific, especially the biologics that you asked about before, testing in animals simply isn't predictive of what's going to happen in a human. Their livers are different, so they're going to metabolize the drugs differently. And so even the variability we see just in humans using different donor pools or single donors, take that to a completely different species. And, just because you don't see toxicity in a mouse or a rat, then that doesn't mean you're not going to see it in a human. And unfortunately, that's what happens 90% of the time, is that something that was not seen in an animal is something that happens once the drug is actually put into the human population, which is unfortunate. So yes, it's a huge opportunity. And we feel that our system in particular has some significant advantages. As I mentioned, that's synthetic hemoglobin that keeps the tissue in a state where it will behave as it would in a living human. And so, for our general first pass assays, we use that 10 donor pool so we can get what we consider a normal human response. But we also have the capability of incorporating cell types from different donors with known, biological or genetic liabilities. And so we can see how a subpopulation may respond differently to a certain drug.
Right. Awesome. Thank you for that. So another question about the specifics of your technology and how it works. Do you use iPSCs?
So I can't really divulge all the cell types that we use. I can say we most often use primary human hepatocytes and commercially available cell lines. In other studies unrelated to what I presented today, we have used iPSCs in our system when we were first developing it and validating it, and just throwing as many cell types at it as we could to demonstrate the benefits of having that synthetic hemoglobin. So, but with the studies that I presented here, 10 donor pool, primary human hepatocytes, and commercially available cell lines.
And would it be possible to incorporate T cells into your model?
Yes, absolutely. Absolutely. So as I mentioned, our system is amenable to anything. So if we were to do that, I would recommend having the 3D culture with the primary tissue of interest, be it so liver, for example, in this case. And then the T cells could initially separately be cultured and then incorporated into the well of the insert. Be they activated prior to that to see if activated T cells have an effect on the tissue that you're growing, or if the treatment that you've performed on the tissue itself results in that immune activation of those cells.
So in addition to the Lena Bio website, which we have up on the screen now, where can folks go to learn more? Are there particular publications you would point them toward? Are you guys planning on presenting at any upcoming conferences?
So we have publications listed on our website, as well as conference presentations that we've given previously. I believe we are planning to attend the Microphysiological System World Summit next year. I believe that's in Seattle in June, if I'm not mistaken. Because we've been in a push for other things recently, so we haven't had a chance to set up any conferences earlier, but I think we'll definitely be attending that one.
Thank you so much, everyone, for your time, and have a great day.